I'd like to present for you our work about a live vaccine against equine influenza based on the cold adapted recombinant strain AHK Otar 6 and 2, 2010. My name is Nurlan Sandybayev. I'm working at Research Institute for Biology Safety Problems, Ministry of Education and Science, Republic of Kazakhstan. The main of activity of our institute are researches for the scientific, technical, and methodological support for biological safety, monitoring of especially dangerous diseases, and development of the diagnostic and therapy and prophylaxis means against special dangerous infection. In this table, we can see that last 3 years, in our institute was developed more than 10 technologies for vaccine against human and animal diseases. The table, a list of vaccines that we developed using recombinant strains. It's first, Cas-Flu-Vac vaccine for H5N1 when we used recombinant virus AS/RG, and Refluvac, you know, is NIVG, TB flu vaccine. This vaccine was developed using influenza as a vector. When we put in genome of influenza 2 genes from tuberculosis, ESAT6 and AG85. This vaccine now in beginning 2 phase of clinical trials. Also seasonal Cas-Flu vaccine and vaccine against brucellosis. This vaccine developed for animals. In this case, we used also influenza as vector, where we put in genome of influenza 2 gene from brucellosis is OMP16 and L7, L12. And of course, a vaccine for equine influenza. So about this one, I will talk today. You know that equine influenza infection is high contagious disease of horses that characterized by lateral inflammation of the upper respiratory tract, total depression in difficult cases pneumonia. You know that currently now 2 types of influenza virus, equine influenza virus. It's first H7N7 and H3N8. The first strain is Miami with Prague, equine influenza virus that was isolated in 1956 in Czechoslovakia. This virus very low happened in the world. The last confirmed outbreak was in 1979. Another strain, Miami, you know, with type H3N8 was isolated in 1963 in Miami. This subtype confirmed all of outbreaks around the world. We also studied about the spread of equine influenza in the Kazakhstan. Our results show that commonly seropositive animals located in north of our country and also in south and east regions with subtype of H3 and 8. In first stage of our work, we constructed a recombinant strain that will be safe, and for this one we used 2 parent virus. So one is first equine Otar. This virus was isolated in Kazakhstan in 2007 and was used as a heme agglutinin and neuraminidase donors. And another virus is Hong Kong. This is vaccine strain with temperature sensitivity and cold adapted phenotype. This strain was used as donor for all of internal genus and will be kindly provided by Research Institute of Influenza, Russian Federation. The next step, to actuality of our wild type virus, we sequencing humoralimmune genes. This phylogenetic analysis shows that our virus belongs to the American lineage Florida, clade 2, and has 99.99% homology to the strain equine Richmond, which has recommended by Officer International Episodes for the production of vaccine against equine influenza. In this slide, you can see a general plan how we construct a recombinant virus. We use a classical method, genetic method. This step includes obtain the specific antiserum, inactivation of wild type virus by UV, mixed incubation in chicken embryos in different temperatures, 32 and 26 degrees, and of course with different amounts of specific antiserum and dilutions. In the end of this work, we will receive a new recombinant strain that we call it AHK Otar 6 and 2, 2010. This strain was temperature sensitivity and cold adapted phenotype. And genome of our new virus have two gene arginine and neuraminidase from our wild type virus in another internal genus from vaccine strain. Okay, in next step, we started our strain for pathogenicity, safety, and stability. Pathogenicity did not cause deaths of chicken embryos during cultivation. And safety, our reassortant was not pathogenic for mice at internal introduction, so it did not cause considerable decrease of body weight and development of the critical symptoms of infection. And stability was estimated by 5, 15, and 12 passage in chicken embryos, and with following PCR and sequence checking. Our result shows that this strain was not changed after 20 passage and keeps TC and cold adapted phenotype. This is picture electron microscopy of our strain after 15 passage. Okay, using our received new strain, we created a vaccine against equine influenza. In first step, we started safety of our vaccine for yearlings. It has been demonstrated that the vaccine was safety for yearlings at internal introduction at dose 9.2 LG AED 15 to milliliters. General clinical condition of the yearlings in both experimental and control groups was normal after 21 days observation. In the upper part of the respiratory tract of the yearling from the experimental group, isolation virus was very low level. The virus was isolated only 50% of animals on first day and 37.5% animals on the day of 3. with very low titer. And noted that all experiments with animals were carried out according to national and international laws and guidelines on animal handling. The protocol were approved by the Committee of the Ethics of Animals, our institute. The next step, we started safety our vaccine on pregnant mares. The test was also safe for pregnant mares. Clinical condition pregnant mares in both experimental and control groups was normal over 21 days of survey. The pregnancies proceeded normally. Rectal temperature of the pregnant mares in experimental and control group did not differ during the observation time. In upper part of the respiratory tract of the pregnant mares from the experimental group, isolation of the virus was less than the yearlings. The virus was isolated only first day, about 20% of animals, and doses between 0.75 and 1. In next step, we started productivity of our vaccine of model challenge. The analysis of data of property showed that vaccine at single intranasal introduction for yearlings provide protection against wild type virus. This protection was caused that intensity and duration of clinical signs, including temperature, reaction, and also virus isolation of animals experiment group significantly lower in comparison with animal on control group. We also studied the duration of protective immune response, not only by single immunization and of course by booster immunization. As shown in pictures, both primes and booster vaccination yearlings generated protective immune response in the last 12 months. After challenging with the wild type virus, the duration of the clinical signs of diseases and duration of the viral shedding were significantly lower in the vaccinated animals than the control groups. The protective immune response was significantly higher in the double vaccination group than in the single vaccination. For example, when double vaccination group were changed at 4 months, 4 months after booster vaccination, no clinical signs of disease were observed in any animal, and viral shedding occurred only in 30% of animals. Double intranasal administration of the vaccines at the interval of 42 days not only provided significant clinical and virological protection against wild type virus, but prevent viral shedding during 3 months after the booster immunization. In conclusion, I'd like that using classical genetic reassortment was obtained new strain AHK Otar 642 2010 with cold adapt and temperature sensitivity phenotype, both virulent and stability, safety. Based on this strain, we created a live vaccine against equine influenza, showing safety to yearlings and pregnant mares. Estimated that booster immunization 42 days after first provide more significant protection against wild type virus as compared to single vaccination. During 12 months, vaccine can be protected animals from the wild type influenza virus. And then about the safety, life science vaccine based on the reassortant strain, which are associated with risk of reversion or reassortment with any wild type virus. In practice, such problems have not occurred during the more than 20 years of positive experience with intranasal live attenuated vaccines among humans in the Western Europe and Russia and other countries. Thank you very much for your attention.
Questions? Okay, thank you very much.
Thank you.